recording, share the screen. Let's get rid of that. Come on. I think I can close that down. So we were talking about the start of microbiology, or what we're covering, and I think we're about on slide 16. So we covered what pathogenic microbes are, and that most microorganisms are not pathogenic. And we talked about what an infectious disease is and what are inherited diseases. Let's look at some microorganisms. Here we're looking at some, and they're small organisms that you need a microscope to see them with. They come in various shapes and sizes, and let's see if I can blow that up a little bit. So there's the size there. Let's put that at the bottom, and there, and this is the smallest one. See if I can scroll down a little bit. There we go, right here. These actually is each of these dots, the green dots, is a cell. And this is a colonial protist, and you'll notice that this is the parent colony here, and inside the parent it's got 6 daughter colonies growing inside of it, and it's going to open up and then release the daughter colonies to go out. There's actually a cell. It might be a white blood cell, a phagocytic cell, and one of the larger cells, and then there's some mold here. The important part is that microbes come in various shapes and sizes. They are small. How small is the average, well, we'll say the average bacteria? About what size is it? Come on, people, you got it right here. How small is the average bacteria?
1 micrometer?
Yeah, about 1 micrometer up to, and usually they're skinnier, but they could be up to 2 micrometers. But 1 to 2 micrometers is your average bacteria, and that's how small they are. Now remember, that's 1 over 10 to the 6 zeros, meaning 1 and then a million zeros, so 1 times 10 to the power of 6, and that's 1 micrometer. So it's 1, what is that, 1/1,000,000th of a meter. Get that smaller. Microorganisms play many beneficial roles to people and to the world's ecosystem. We mentioned that they decompose organic waste, like when you flush the toilet or flush something down the drain. The sewer plant, treatment plant, it uses microorganisms to decompose the organic waste. Microorganisms are producers in the ecosystem on the planet Earth. Many of them engage in photosynthesis or chemosynthesis, making glucose from carbon dioxide and water. Microorganisms are involved in the nitrogen cycle. We talked about that yesterday. And they're involved in other cycling of nutrients in the planet Earth, such as sulfur and carbon and what else, phosphate. We'll talk a little bit more about some of those, just a little bit. Microorganisms are important in producing things for us, such as industrial chemicals, like the ethanol you put in your car, that's made by microorganisms. And it's true, it comes from corn. That's what the microbes digest to make the ethanol. But it's the microbes that are making the ethanol. And many other industrial chemicals are made by microorganisms, like acetone. Some insecticides are made by microorganisms. And we'll talk about one later on, just in a little bit. All fermented foods are made by microorganisms, and that includes things such as vinegar, all cheeses, most leavened bread. Unleavened bread does not use microorganisms, but leavened bread, most leavened bread is made by microorganisms, and it's fermented to make the leavening. Beer and wine also made by microorganisms. Many of the products used in manufacturing are made by microorganisms. One example is cellulase. All of the early antibiotics, and even some of the antibiotics today, are made by microorganisms. Some nowadays are totally man-made, and we don't use microorganisms to make them, but some still are, like penicillin G. Other products used in medicine, like insulin, is made by microorganisms. They took the gene from humans out of a human cell and put it into a bacteria, and I think it's E. coli, but I'm not sure which species is making it. And now that microorganism is making the human insulin, and that is how we make insulin today. Microorganisms are also important in biotechnology and genetic engineering. Biotechnology is actually the use of microbes and other organisms to produce foods and products for human use. And this is centuries old. People have been using biotechnology for centuries. It's just that there's a new form of biotechnology using genetic engineering that is new. And that's usually what we think of as biotechnology. But the making of like sauerkraut and actually all types of cheeses is using microbes to make products like food, which I'm talking about all of them food, like sauerkraut and kimchi and cheeses. And that is biotechnology. And people have been doing this for centuries. Let's see. The production of beer and wine has been going on for more than centuries. It's been, gosh, over 2,000 years, maybe over 10,000 years. Okay, so biotechnology is at least that old. And I'm trying to remember, I think the Egyptians made wine before they made bread, and we'll talk a little bit more about that. Genetic engineering is a new technique of biotechnology, and this began, I don't know, 1980s or 1970s. Some applications of genetic engineering is the production of a variety of proteins from bacteria and fungi. Move that over there. Such as the making of vaccines today. Most of that is done by using microorganisms. Trying to think, is there any vaccine that's not done by using microorganisms? And I sure can't think of any. The making of a variety of proteins, such as enzymes that we make and use. And then various antibiotics are made by both bacteria and fungi. Gene therapy, another application of using microorganisms. Gene therapy is when somebody is born with a defective gene, and then they insert, using a microorganism, the correct gene into that patient so that they then receive gene therapy to correct their genetic defect. Any questions about any of that? Gene therapy in people has been best studied and used, or maybe, yeah, best used by correcting children who are born with a defective immune system. And they have a defective gene which doesn't allow their immune system to function fully, and then we can give them the correct gene, which is a human gene, but we put it in a microbe, usually a virus, and then insert it into their immune system cells and then inject those cells into them. Anyways, that's gene therapy. There are some other applications of gene therapy, but that's the one that's been used the longest. There are genetically modified bacteria which we use to make products for us, as well as protect crops and other things, like protecting crops from insect and from freezing. Actually, the insulin that we talked about earlier was a genetically modified bacteria. They put the human insulin gene into the bacteria and then grew up the insulin in the bacteria. All right, any question about any of this? Microbes are important in human welfare. We use, or I should say the ecosystem uses, microbes to recycle various nutrients around on the planet, such as carbon, sulfur, and phosphorus. So these are three I really haven't talked much about. And then we did talk a lot about nitrogen, how the nitrogen cycle happens because of microorganisms. Now, carbon is cycled in the ecosystem partly by microorganisms, but carbon also moves around in the ecosystem by other means, not using microorganisms. And then microorganisms are extremely important in recycling nutrients. When an organism dies, like a tree, it will be decomposed by fungi and microorganisms, meaning mostly bacteria and archaeobacteria. And I said fungi because most fungi are microorganisms, but the mushrooms are not, and they can help recycle a tree as well. The nutrients, pull them out of the tree and then release them so that other organisms can use them, including other plants and animals. And the recycling of carbon, nitrogen, sulfur, and phosphorus in the ecosystem happens by microbial mediated biochemical cycles, and these cycles are needed to support life on Earth. If we didn't have these cycles, the carbon, the nitrogen, the sulfur, and the phosphorus would all accumulate in one form and wouldn't be converted to other forms. And fortunately, the microorganisms recycle those nutrients, supporting life on the planet Earth. The first person to study the microbial ecosystem, where the microbes were responsible for mediating the biochemical cycles of the nutrients on the Earth, was studied by Winogradsky and Bajorink, and that happened between 1880 and 1892. You don't need to know the date, but you should know that Winogradsky was the primary person who discovered that, and the first to show that nutrients and elements are recycled on the planet Earth by microorganisms. And if you've ever heard of the Winogradsky column, we'll be talking about that a little bit later in the term. He used that column to study the recycling of nutrients, primarily sulfur. What was the other one he was studying? Nitrogen? I think it was sulfur and nitrogen in the Winogradsky columns. All right, any question about any of that? Microbes are also important to human welfare, and that is we can use them in bioremediation. Bioremediation literally means life and to remedy, where we can use living organisms to deal with an environmental problem, such as a pollutant. Like right here, these gentlemen are spraying a microorganism on this oil spill to have the microorganisms degrade the oil. And we use that to have the microorganisms degrade our sewage and various pollutants such as oil, as well as mercury. Now, mercury cannot be totally degraded into some other element, but we can convert one form of mercury into a less toxic form of mercury by using microorganisms to treat or remedy the pollutant. Any question about any of that? We can also use microbes to make biological insecticides, and this can be an alternative to a man-made chemical pesticide. Generally speaking, the biological insecticides are less toxic to humans, or even sometimes non-toxic. For example, Bacillus thuringiensis is used to prevent damage to crops and disease transmission to crops by killing various insects and preventing the insects from spreading disease. Bacillus thuringiensis is a bacteria where the infection is fatal to many insects, but Bacillus thuringiensis is harmless to humans, other animals, and plants. And as early as the 1960s, I don't remember exactly the year, I was just a kid at the time, very young, many were shortly born, but I remember my father would take Bacillus thuringiensis and put it on the apple trees, and if done appropriately, the Bacillus thuringiensis would prevent the apples from getting worms. The trouble was, he did this in western Oregon, which has a lot of rain, and he didn't know that you should treat the apples every time after it rains, because the rain washes the Bacillus thuringiensis off the apples, as well as you really need to treat the apples about every 2 weeks, which is how often you need to treat it with the chemical insecticides. And he was far too lazy to do it that often, and so our apples did have worms, but they had fewer worms than if he hadn't treated them. Any question about any of that? All right. Any question about how microbes are important to human welfare and to the planet? We'll talk about in a little bit how microorganisms are also important for helping us to digest our food, that all animals use microorganisms to help digest their food. And some animals, like cows, would not be able to digest their food if not for the microorganisms, meaning cows need the microorganisms to digest grass. We don't tend to eat grass, and so we can partially digest the food, but we do have help from microorganisms in our intestines helping us digest our food. If there's no questions, I want to move on to another topic, the naming and classifying of microorganisms. And this topic was begun by this gentleman here, who started it in 1735. You don't need to know the date, but he started the scientific naming and classification system. And here we're talking about the scientific naming system, which is called the scientific nomenclature. And who is this? Who knows that? Come on, I know somebody does it because I saw that some people had done the
It's
book.
Carolus Linnaeus.
Yeah, Carolus Linnaeus, and his name, before he Latinized his own name, he liked Latin names so well he changed his own name, but it was something like Carl Linné or Linnea, uh, from one of the Scandinavian countries. I think it was Sweden, but I'm not positive. I don't remember. But he liked Latin names so much he changed his own name and Latinized it. And what he did was he gave each organism a scientific name, and it was based on two names, a genus name for the genus and then the specific epithet for the species. So all species have two names. For example, humans are called Homo sapien, Homo being the genus, meaning man that walks on 2 feet, and sapien is the specific epithet, meaning wise man. And that is our species name. And this system is called the scientific nomenclature, also called the binomial nomenclature, because all species have two names, the genus and the specific epithet. Any questions about any of that? This nomenclature is still used in science today. We did change the classification system, like he was classifying everything into animals and plants, and we'll talk a little bit more about that. We have more divisions than that. But the nomenclature is still used today. In his binomial nomenclature system, all names are italicized or underlined to let people know that it is a scientific name. So if you're typing it, you italicize it. If you're writing it and you can't write in italics, which I certainly can't, you underline it. The genus name is capitalized and the specific epithet is lowercase. All scientific names are Latinized and used worldwide, meaning the same species name is used wherever you are in the world, whatever your language is. So everybody in the world uses the term Homo sapien. Some example, obviously Homo sapiens for humans, Escherichia coli for E. coli, Staphylococcus aureus for Staphylococcus aureus. After the first use, the scientific name may be abbreviated, and you abbreviate the genus name to the first letter and a period, and then you put out the specific epithet, like Staphylococcus aureus. The first time you use it, you put out the full name, and then you can abbreviate it as S. aureus for the rest of your paper. E. coli is the abbreviation for Escherichia coli, and this one is so common, and everybody knows what E. coli is, that even scientists will occasionally only say E. coli, meaning that one is so common, it's much more common than Escherichia coli. Staphylococcus aureus, for your information, is found on, or can be found on human skin. Actually, hopefully it's not found on your skin, but it can be found on human skin. And E. coli is found in the intestine, particularly the colon. Where do I have that? I thought I had something in here. The name, oh, there it is there. Forgot to mention this here. The names that are chosen to be either descriptive, like E. coli. The coli is named after descriptive, that it's found in the colon, so it's given the name E. coli. Or it can, the name could be to honor a scientist, like Escherichia is honoring an early microbiologist who I think was Esther. A Homo sapien is totally descriptive. Homo means man who walks on 2 feet, sapien, wise man. Staphylococcus aureus is totally descriptive. It literally means aureus, golden, because the cells are yellow in color. And then Staphylococcus is a ball-shaped cell that grows in a cluster like grapes, and that's what Staphylococcus means, a ball that grows in a cluster like a grape cluster. All right, any question about any of that? All right, so before we talk a little bit further about the classifying of microorganisms, we need to address how we group organisms, and this is the classification of all living things on the planet Earth. The first division or classification is called the 3-domain system. So all living things are divided into one of 3 domains. The 3 domains are the domain bacteria, the domain archaea, also called the archaebacteria, and then the domain eukarya. How we know what an organism, which domain an organism falls under, is you simply sequence the organism's DNA, and then we find its domain by the domain that has the sequence of DNA, meaning all organisms in the domain bacteria have a similar sequence of DNA. And all of these organisms are more similar to each other than an organism in another domain. So the domains are based on the genetic data of an organism. And there's no argument as to where a species falls, because you simply sequence the organism's DNA and determine what domain the organism belongs to by its DNA sequence. Is that clear? Okay, no questions, so let's move on. The 3-domain system is sometimes called the tree of life because all living things branch off from one another in a 3-branching system that reminds people of a branching tree. Of course, the tree is got 3 trunks, but it is a 3-branching tree of life. The domain eukarya can be further divided into kingdoms, and that would include, so this domain has 4 kingdoms: the kingdom Plantae, the plants; the Animaliae, the animals; the fungi, the fungus; and Protista, the protists. The kingdoms generally are organisms that are similar to each other, like the plants are organisms that do not move, and most of them engage in photosynthesis, although there are about a dozen plants which are parasitic and they're not photosynthetic, but they have plant-like structures and cell walls and stuff like that, compared to the animal and the fungi. And all of these 3 kingdoms are unique, and every organism within that kingdom is similar to each other. The kingdom Protista is separate, and it is a eukarya, which is not one of the other kingdoms, meaning not a plant, not an animal, and not a fungi. So the Protista is a kingdom which is unique in that it is not monophyletic. It's a collection of all other eukaryotes that are not plants, animals, and fungi. Any question about any of that? Okay. So, like I said, we have the classification of living organisms, and we classify all living things into 3 domains: the bacteria, the archaea, and the eukarya. You then can divide the domains into kingdoms, and the bacteria, generally speaking, only have 1 kingdom, so it's given the same name as the domain bacteria, and then you divide that into the kingdom bacteria. The same with archaea. It only has 1 kingdom, so the domain archaea also has a kingdom archaea. And then the domain eukarya has 4 kingdoms: the plants, the animals, the fungi, and the protista. Any question about that? You can divide things further. We're not going to talk about further classification. Let's see now, what's after kingdom? King Peter's. So phylums would be the next. We're not going to talk about the other further classification. When we're talking about the 3 domain system, you should note that the domains have a cell type. The bacteria have the cell type prokaryotes, meaning they have prokaryotic cells. The archaea also have prokaryotic cells. And then the eukarya have eukaryotic cells. And this is the way your textbook author talks about it, that the domains have a cell type. Some people call the cell type a super kingdom, and your textbook author doesn't use the term super kingdom. I'm just mentioning it because some people do, and if you run into it now, you've been exposed to it. So the cell type is the same thing as the super kingdom. It's just that your textbook author doesn't have a super kingdom. All right, any question about the cell type? If not, let's talk a little bit more about the domain bacteria. The cell type is prokaryotes. The domain bacteria has peptidoglycan in their cell walls. The cells can multiply by binary fission. That's the majority of bacteria. Binary fission is simply where the cell divides in 2. So this cell, when it gets ready to divide, will simply split into 2. The bacteria can get for their energy needs. They can use an organic chemical, such as glucose, and that's what we do, using organic chemicals. But some bacteria can use inorganic chemicals, and that could be something like nitrate or sulfate or hydrogen sulfide gas. And we, of course, can't do that. And some of the bacteria can get their energy needs by photosynthesis, and that would be a photosynthetic bacteria. Any question about any of that? Most of the bacteria are free living decomposers or primary producers, meaning they decompose things or they can make their own food by chemosynthesis or photosynthesis. That's what a primary producer is. They can make their own food. Some of the members of domain bacteria are parasitic, living off of another organism. And some of the bacteria are pathogenic. And that is a bacteria that can cause human disease, like Staphylococcus aureus is a pathogenic bacteria. Some of the characteristics of archaea: all of the cells are prokaryotic. Does anybody know, do the archaea have peptidoglycan in their cell wall? That's a yes or no.
they do not.
No.
Yeah, that is correct. The archaea do not have peptidoglycan, only the bacteria do. Some of the archaea are found in extreme environments, And that is where we first found the archaea. That includes organisms living in methane gas environments with high methane gas, and that would be the methanogens. Archaea living under high salt concentrations, like in the Dead Sea, and that would be extreme halophiles. And then there's extreme thermophiles, archaea, living in very hot environments. But when we look further, we can find the archaea in all of the Earth's biomes. They are actually a significant number of the organisms in the soil and in plankton in the ocean. So they are important for making up organisms on the planet, and you can find them in all of Earth's biomes. Does anybody know, are any of the archaea pathogenic to humans? Once again, that's yes or no, people.
No.
Yes. None of the archaea are pathogenic in humans, meaning there are no archaea that cause human disease, as far as we know. So for that reason, we will not, after this lesson, be studying the archaea very much. Occasionally, we'll briefly discuss them, but we're not going to talk about the archaea very much in this class, because the focus of this class is to talk about the pathogenic organisms, meaning the pathogenic bacteria, and that does not include any archaea. So we will not be focusing in on the archaea just because of what this class is focusing in on. And we're discussing the pathogenic bacteria because most of you are going to be working as a healthcare provider or working in a clinic or something like that. I forget what you call that. You're going into the biomedical sciences, and so the focus of this class is going to be on the pathogenic bacteria. And only the bacteria have pathogenic prokaryotic cells. We will talk a little bit about the eukaryotic cells, which can cause human disease, and usually we call them parasitic organisms, and that would be something like malaria. What's another one? What's another parasitic disease caused by a protist? Entamoeba histolytica can cause amoebiasis, diarrhea, severe diarrhea, for example. So we'll also be talking about some of the eukaryotes, which can cause human disease, and then the bacteria that cause human disease. And the bacteria, we call them pathogenic organisms or pathogenic bacteria. For the eukaryotes, we tend to call them parasites. And you could use the term of pathogenic protist, but most people call them a parasitic protist. All right, any question about any of that? So the archaea are interesting and fascinating, and I wish we had more time to talk about them, but we're not going to focus on them. Let's talk a little bit about the characteristics of the eukarya. I mentioned that we can divide the eukaryotes into 4 kingdoms: the plants, the animals, the fungi, and the protista. It's important to note that all of the eukaryotes lack peptidoglycan in their cell wall, and of course animals don't even have a cell wall, so that only the bacteria have peptidoglycan. Let's talk a little bit about the kingdom fungi of the eukaryotes. Fungi is plural. The singular term is fungus. You don't need to really know. You can find chitin in their cell walls, but they use organic chemicals for their energy needs, And that's similar to us. We use organic chemicals for our energy needs. The fungi include the multicellular molds and the mushrooms, which are multicellular. They consist of masses of mycelia, which are composed of filaments called hypha. And there we're seeing a hypha right there and right there. The fungi also include the unicellular yeast. Some of the yeasts in particular are human pathogens, but all of the fungi are either decomposers, most of them are decomposers, and some of them are human pathogens, like Candida albicans, causing human disease, a number of human diseases, but yeast infections and what is that called on the skin? Drawing a blank here. Jock itch around the genitals, but on the skin, what the heck is that called? I'm drawing a blank, sorry. Candida albicans causes skin disease and can grow on the skin and drawing a blank, sorry. Diaper rash in babies, but I'm drawing a blank. What is it called, people? Somebody probably knows. What is a yeast infection on the skin called? Nobody's going to state. Anyways, I can't remember. I'm just drawing a blank here. It'll come to me. I'll just have to think about it. Let's talk about the protists, the protista. We can divide these into three types. There's the plant-like protists, and these include the algae. They all have cellulose on their cell walls, similar to plants. They engage in photosynthesis to get their energy needs. They produce oxygen. They typically are green, but you can have brown algae and red algae. There are other colors. And they do make organic compounds, largely glucose, by photosynthesis. And then there's the animal-like protists. Those are the protozoa, including the amoeba, the paramecium, the euglena. All of these organisms absorb or ingest organic chemicals from outside, like right here, that can close up from the pseudopods and be incorporated inside, and then the organism can ingest that food on this side. The protozoa may be motile with the pseudopod, shown here, cilia, which actually this organism has, and flagella. Some of the protozoa can be parasites, like the Entamoeba histolytica, causing amoebiasis or malaria. So some of the protozoa are parasites of humans, causing disease. And then the third category of the protists are the fungus-like protists, and they have properties like the fungi. Any question about that? All right, let's talk a little bit about the kingdom plants. You can divide the kingdom plants into the mosses, the ferns, the conifers, and the flowering plants. All plants are multicellular, meaning more than one cell makes a plant. We won't be studying plants because they do not cause human disease. Let's talk a little bit about the kingdom animal. You can divide the kingdom animals into the sponges, the worms, or scientific term, the helminths, the insects, and the vertebrates. All of the kingdom animals are multicellular, meaning that an organism has more than one cell. Now, we won't be talking much about the kingdom animal except for the helminths, because sponges and insects and vertebrates, except for the animals, do not directly cause human disease. Insects can bite us and then give us disease, but the insect is a vector bringing the disease-causing organism to us, like when a mosquito brings malaria to man. The disease isn't caused by the mosquito, it's caused by the malaria parasite in the mosquito. We will be talking a little bit about the helminths, the worms, because those do cause human disease, and that would be a worm infection. Any question about any of that? So when we're talking about microorganisms, we need to just discuss what do we mean by what falls under the umbrella of a microorganism. Okay, all of the bacteria are microorganisms, all of the archaea. Viruses and prions are also considered microbes, and they're not exactly an organism because they're special. They don't have cells, so they're not an organism. But they are a microbe, and they do cause human disease, so microbiologists do study them. And certain of the eukarya also fall under the umbrella of a microorganism. All of the protozoa, and they're singular celled, and you need a microscope to see them. The molds and the yeast are microorganisms. Some of the algae, if they're microscopic, they are a microscopic algae. We consider them a microorganism. That does not include the algae that washes up on the Washington coast or the Oregon coast. Those things are very large, like the brown kelp. And those are algae, but they're not microorganisms. So some of the algae are microorganisms and other algae are not. The helminths are considered microorganisms, but worms aren't microscopic, right? So why do we consider a worm a microorganism? Well, the helminths are special. They are multicellular animals, and many of them are parasitic. The parasitic flatworms and the parasitic roundworms, causing disease in humans. And generally, something that causes disease in humans is something that a microbiologist studies. So microbiologists have included the helminths in the term microorganism. And they are, not strictly speaking, microorganisms when you're looking at an adult helminth. Although there are a few species which are microscopic, most of the helminths are not microscopic. But the life stage of the worm, which is infective to humans, is microscopic. And it's usually the fertilized egg, which is the infective stage of the worm, which infects the human. And this life stage is microscopic, and it's the one causing disease in humans. And that's why microbiologists study helminths, even though the adult worm usually is not microscopic. Its fertilized egg is. And that's the stage which is infective to humans. All right, any question about any of that? Let's talk briefly about viruses. We will talk further about viruses near the end of the term. I think it's the second to the last chapter. But we just need to talk a little bit about them. Viruses are acellular, meaning they lack cells. They do have a genome that consists of either DNA or RNA, but not both. And that molecule is the core central molecule of the virus. That core molecule is surrounded by a protein coat, so all viruses have a core genome that's surrounded by a protein coat. And then some viruses have a lipid envelope around the protein coat. Not all viruses, only some of them. And when do viruses replicate? Viruses replicate only when they are inside their host cell. So a virus infects their host cell, gets inside of it, and then the virus can replicate. A virus cannot replicate outside its host cell. Viruses are not part of the three-domain system because they're not cellular. They have no cells. Any question about that? We'll talk more about viruses near the end of the term. This table is comparing the three domains of life, the bacteria, the archaea, the eukaryotes, looking at the cell type, their chromosome, their ribosomes, and their cell walls. You do find differences between the domains in these characteristics. Not all of them are different, like the ribosomes of bacteria and archaea are the same size, but some of them are different. All right, any question about the three domains of life and microorganisms? If not, let's go on and talk about a brief history of microbiology. When we're talking about the history of microbiology, you should realize that we start that when microbiology began in 1857, and this will incorporate the early age of microbiology, which is often called the golden age of microbiology. And that happened between 1857 and 1914. And you don't need to know these dates. The golden age of microbiology was a golden period because everything they looked at was something new, a new discovery. And that's kind of why it was called the golden age of microbiology. The early history of microbiology and the golden age of microbiology largely concerned a debate over one thing: whether life could spontaneously generate or whether life came about from the theory of biogenesis, meaning life came from pre-existing life. And there was a debate about that, especially in microbiology, but actually that happened in biology also. And here's a little synopsis of what we're going to be talking about. We won't talk about all these things, but the history of microbiology, which actually begins before microbiology was discovered, and then the golden age of microbiology, and then the age of microbiology after them. So when I'm talking about the history of microbiology, it begins in ancient times, and I'm going to be giving it in dates just because it's easier to talk about something by giving it a date. But you do not need to know any of the dates except for this first date. You do need to know that over 3.5 billion years ago, the ancestors of the first bacteria were seen on the planet Earth. And this was the first life on the planet Earth that we know of. So over 3.5 billion years ago, we can find fossil records of ancestors of present-day bacteria, meaning they weren't bacteria back then, they were the ancestors of bacteria. Any question about that? And this picture here is showing you some of the fossils. Not 3.5 billion years, but close to it. This is 3.46 billion years. After that, history of microbiology takes a big jump in years, but 4,300 BC, the Babylonians recorded the first observation of microorganisms. And that is, they wrote down tablets on their clay tablets, recipes for making beer. They didn't know it, but microorganisms were responsible for making beer. And then 600 AD, the Mayans recorded the fermentation of a beverage from cacao, which is chocolate. We would call it chocolate today. They made a fermented beverage of chocolate, and that would be the second thing I'm going to talk about in the history of microbiology. Although you don't need to know these dates, you should know that I may hold you responsible for knowing the relative order of things. Like the Mayans could not have recorded the fermentation of chocolate until the very first recording of the fermentation of beer, which couldn't have happened until after we had a fossil record of the ancestors of the first bacteria. So I may hold you to the relative order of the topics, but I'm not going to ask you. In 600 AD, the Mayans recorded the first fermentation of a chocolate beverage. Any question about any of that? All right. So the next important event in the history of microbiology was in 1590. You don't need to know the year, but Janssen developed the compound microscope. And that was really important because until the microscope was invented, we would not be able to see microorganisms. Surprisingly, nobody saw cells initially when Janssen made the microscope. It wasn't until 1665 that Robert Hooke reported seeing the first cells. What he did was he took shavings from cork and from other plants, and he could see the very first cells. When he looked under the compound microscope, he saw little boxes of plant cells. And thereafter, people would look at more and more organisms, and from that, they came to the conclusion that all living things are composed of cells, and that was the beginning of the cell theory. So the cell theory started in 1665, but it did not come about until many years that people would keep on looking at things and seeing cells, and then they realized, you know, maybe all things are made of cells. And that's the cell theory, that all living things are made of cells. And that was written by Virchow many years, I think it was over 100 years after we saw the first cells. Now let me state that in 1665, and for nearly 100 years, people could not see cells in animals because the microscopes were so blurry, we could not see cells in animal cells. But when they looked with the early microscope at plants, we could see cells. Why do you think that the early microbiologists or early biologists could see cells in plants but not in animals? What do plants have that animals do not?
A cell
Come
wall.
on, people, what do plants have that animals do not?
A cell wall.
Yes, the cell wall was very thick and very rigid, and even with the very primitive microscopes, biologists could see cells in plants because of the cell wall. But when they looked at animal tissue, they couldn't see the cells because the microscopes were not good enough to see the cell membranes. They're much thinner and fragile and flexible compared to the cell wall. And so the early biologists could not see cells in animal tissue. It had to wait for something like 100 years or more before they developed better and better microscopes until they got one good enough that it was powerful enough and clear enough that somebody could see cells in animal tissue. So initially we could not see cells in animals because the microscopes just were not good enough. Now these above discoveries, Jensen developing the microscope and Robert Hooke discovering cells and plants, were not specifically microbiology. Robert Hooke was seeing cells and plants, not in a microorganism. So the above is not strictly speaking microbiology, but all of these had to happen before we could see the first living microorganism. And I will talk about that. I'm going to move forward because I'm talking about that now. The first microorganism was seen by Antony van Leeuwenhoek, and the reason why he probably saw the first microbes was he was an amateur microscope builder, and he ground his own lenses, and he was the best lens grinder and microscope builder of his day. So he had the best microscopes, and he would look at all sorts of things, and generally whatever he looked at he could find microorganisms, like scrapings from his teeth when he looked in rainwater, peppercorn infusions, and even the water from the River Thames, which was the river flowing through London. Any question about any of that? So he's the first one who saw microorganisms, which happened much later than Robert Hooke, almost 100 years later. Not quite, but almost. When we're talking about the history of microbiology, it's important to realize that the early microbiology was concerned with the generation of life. There were two competing hypotheses for how life originated. And this was from about 1600 to the 1800s, sometime in the 1800s. There were two competing hypotheses to explain how life began. The first was spontaneous generation, that life arises out of non-living things spontaneously. A vital force comes into something and then, I guess, changes it, and then something comes alive. And that was the theory of spontaneous generation. The competing theory was biogenesis, that living forms, new living forms, arise from pre-existing living forms. For example, it'd be very obvious that a baby comes about from his parents or her parents. Any question about that? So those were the two competing theories about how life originated. Now, over a century ago, beliefs included things like if you threw a log into the fire, sometimes a newt would come out of the fire, and people would say, aha, spontaneous generation of the newt. You throw the log into the fire, and the wood and the fire together spontaneously generated the newt. And that was one of the observations they made for spontaneous generation. There were some other, like flies could emerge from manure, and maggots could arise from decaying corpses. These were all things that people said were spontaneous generation. Most were logical, but none of them were scientific. Some of them were even amusing, like they believed, or many people believed over 100 years ago, that sweaty underwear put in a jar with wheat in the bottom of the jar could give rise to mice. In reality, what happened was the mice got trapped in the jar and couldn't come out, and they got in the jar because they were going after the wheat. All right? It wasn't because the sweaty underwear were in the jar with the wheat and then the mouse spontaneously generated. And you can read about that one right here. I'm not going to cover it, it's just amusing. The first evidence for or against spontaneous generation came in 1668 by Francesco Redi, and he gave the first experiment to show that spontaneous generation did not happen. And surprisingly, he also gave the first controlled scientific experiment. So he's the first person to write up a controlled scientific experiment. What Francesco Redi did was he got 3 jars and left them unopened without a lid and put meat in the jars. And then he got 3 jars and he completely sealed them. And he got 3 jars and he covered them with a fine wire mesh net. And the reason why he had this wire mesh was when he completely sealed the jar, people argued, you sealed it so the vital force could not get into the meat and cause spontaneous generation. That was the argument they used. And so he had a jar with meat in it, but he just put a wire mesh over it. And that way the vital forces could get in to the jar. And what he saw in the completely sealed jar and the jar of meat that was covered with the wire mesh, that no maggots occurred in the meat. But he saw the maggots appearing in the meats that were left open to the air. And he hypothesized that the maggots came about from flies which flew into the open jar and laid the eggs on the meat. He didn't actually prove the later point, but he hypothesized that. But the point is, he showed that maggots could not spontaneously generate in these jars of meat. And so his results did not support the theory of spontaneous generation. And after his day, people did not believe that large living organisms, like a maggot, and that's large compared to a microscopic organism, could not spontaneously generate. But people still argued for the spontaneous generation of microorganisms. And you can read a little bit about that here in that slide here. I'm not going to discuss it. We talked about Antonie van Leeuwenhoek being the first person to see microorganisms, and he lived from 1673 to 1723, so something like 1690 is when he had his first microorganisms. And Antonie van Leeuwenhoek was such a good microbiologist and such a good microscope builder that he saw and recorded cells so small, right here, drawings of bacteria, that the microscopes of his day could not see these cells. And it wasn't until 100 years later that the microscopes improved, and then other people confirmed the existence of bacteria that Antonie van Leeuwenhoek saw and recorded and drew 100 years earlier. So he was such a good microbiologist and such a good microscope builder that he could see a cell that other people couldn't see for about 100 years. So that tells you how good Antonie van Leeuwenhoek was. And when you look at these drawings here, you can see that these are clearly bacteria that he was seeing. All right, let's talk a little bit about biogenesis. That theory was stated and written up by Rudolf Virchow in 1858, but people were beginning to believe in it before he published his writing that all living things come about from something that was already alive, meaning the theory of biogenesis. It wasn't until Louis Pasteur came along that the theory of spontaneous generation was disproved, and therefore Louis Pasteur confirmed the theory of biogenesis. What he did was Louis Pasteur demonstrated that microorganisms are present in the air. And then from the air, they can contaminate something that was previously sterile. It wasn't from the vital forces coming in and causing microorganisms to spontaneously generate. Louis Pasteur also showed that microorganisms could be destroyed by heat. So you could destroy the microorganisms in the air by heat. And he made his ingenious, famous swan neck flask experiments to disprove the theory of spontaneous generation. What he did was, he got a flask and he poured media in it, and then he melted the glass at the top and made what's called a swan neck. And then he sterilized the media on the inside of the flask by heating it up and then allowed it to cool. He found that no bacteria, other microorganisms grew in this flask even though it was open to the air. But the microbes from the air came and settled right here by gravity. Because of gravity, the microorganisms never went up here and then down. He could get the microorganisms to grow in the media if he put the media and flushed it and brought it down here and then brought this back into the media. Anyways, the swan neck flask proved that the spontaneous generation of microorganisms did not occur, and therefore living organisms were happening by the theory of biogenesis. Any question about any of that? All right, I think I'm going to stop here and we will continue here next time, and I'll see you at 6:30 in the lab. Are there any questions? If not, I'll see you at 6:30.
I had a question.
Okay, go ahead and ask your question.
Could we have you post the PowerPoints from Tuesday and from today?
I can post them from Tuesday, but I do not have them from today, so you have to wait until this lesson is finished. Okay?
Okay.
Remind me in the lab, and I'll do that in the lab.
Okay, thank you.
All right.